Good morning, students. Today's session we are dealing with the absorption by roots, which is one of the physiological process we can see in plants. So what is meant by plant physiology? It's a branch of biology that deals with the function or metabolism of the plant parts. Okay, what are the different functions we can see in plants? Yes, first one is absorption, next is conduction, then transpiration, transportation, photosynthesis. These are some of the physiological process that takes place in the plant. So today's session we are dealing with the absorption by roots, how roots are absorbing these water and mineral salts. Come on, tell me, what are the functions of the roots? You know it already. So, one of the main function of the roots is to fix the plant in the soil. So, fixation. So, one of the main function of the root is fixation. So, second function of the root is absorption. And the third function of the root is conduction. So these are the three main functions of the root system. So first one is absorption. So only roots can absorb the water and mineral salts from the soil. Next, you can see that fixation. Without the help, you just you cut the shoot system of a plant. You can see immediately the shoot system is falling down. So what fixed this in the soil? Yes, root, widely spreaded roots that fix this plant in the soil. Next one is conduction, so that water and mineral absorbed from the soil has to conduct to the whole parts of the plant. Mainly it has to reach in the leaves for the process of photosynthesis. So these are the three main functions of the roots, that is absorption, then fixation, conduction. These are the three main parts. Now let us see what is the need of water and mineral salts for the plants, or why a plant needs water and minerals. So there are four things are there. First one is for photosynthesis. Why water is needed? First one for photosynthesis. As all of you know that water is one of the raw material for the photosynthesis. If photosynthesis wants to take place, water is must. So, from where this water is getting? Yes, from the soil. Which part will absorb this water? Yes, that is roots. So, here you can see that this water is very much needed for photosynthesis, which is one of the raw material for photosynthesis. Next one is for transpiration. What is meant by transpiration? Yes, the loss of water from the aerial parts of the plant is called as transpiration. So here you can see every day there is a large amount of water is lost by the plant due to transpiration. And by this transpiration you can see the surrounding air get cooled. So the temperature will come down. So for this purpose also water is needed. And third one is for transportation. You can see that transportation of substances in water soluble form only will take place in the plant body. So if it is in solid form, that won't be taking place. So transportation of this, especially you can see that the food materials which is prepared by the plant, it has to reach all part of the plant body. So there the liquid medium is necessary. So here also for transportation also water is needed. And fourth one for mechanical stiffness. Fourth one for mechanical stiffness, and you can see water provides turgidity to the cells. So you can see that, so that plant cell become turgid, then water enters into the cell, and you can see that the plant will become stiff in nature. Otherwise it will be drooping down. If water is not there, you can see that plants will be drooping down. So water is necessary for mechanical stiffness also. So these are the four reasons why water and minerals are needed for the plant. First one is for photosynthesis. Next one is for transpiration. Third one is for transportation, and fourth one for mechanical stiffness. And this, what are the mineral nutrients required by the plant is absorbed by the roots, and these may be in the form of salts or in the form of some ions. And why this is needed? Yes, this is needed for the synthesis of, or you can see that synthesis of variety of enzymes will be produced in the plant body. For the synthesis of all these things, what is needed? Yes, these ions or salts are needed. And now let us see what are the characteristics of the roots for absorbing water. So, first one is that a huge surface area is needed by the plant. So, a huge surface area is needed by the plant. How a huge surface area? Yes, it is by the roots and by the root hair. You can see if you uproot a plant, gently you uproot a plant and wash very gently it with water. You can see that the root will be like this, and you can see the branches of roots like this if it is a taproot system, and still secondary roots, tertiary roots like this, branches, etc. You can see, and each one's end region fine hair-like structures you can see. Here also, there also you can see fine hair-like structures like this. What is this called? What is this hair-like structure called? Yes, these are called as root hairs. So, these root hairs provide a large surface area for the roots so that absorption will be easier. Okay, so the first feature is what? Surface area of the root should be huge. So, in each plant you can see this very small outgrowth called as root hairs are present. So, root hairs inhabits for the huge surface area for the plant. Second one is that root hairs contain cell sap at a higher concentration that of the surrounding water. So, root hairs, these root hairs which are minute structure like this, it contains cell sap. What is meant by cell sap? The liquid or fluid medium which is present inside the cell, that is cell sap, and it is in a higher concentration than the water which is present in the outside cell. So you can see it will be all in a higher concentration so that it will be easy, water can enter into it. So like this here you can see inside the roots the cell sap will be in higher concentration. So some solute will be dissolved in that one, that will be in higher concentration, and so water will be inside the cell sap. Water's concentration will be in low range so that from outside it can enter easily. That is the second reason. And third one is that root hairs have thin walls. So you can see that plant cell, the cell wall is made up of what? Yes, cellulose. And here in plant cell the cell wall is a characteristic feature, whereas in animal cell wall is absent. And here you can see that it have these root hairs This is the epidermis of the root, and I am showing here a fully grown root hair like this. So here small outgrowth you can see, here still again you can see small outgrowth. This is the epidermis of the root. Okay, so in this root hair, it has a thin cell wall, two membrane you can see in plant cell. One is the outermost outer cell wall, and second layer is called as cell membrane. So cell wall is present as well as cell membrane is also present. So the next feature is that it containing a thin cell wall. So that cell wall, you know, the features you have studied last year. What is the specialty? It is freely permeable. It allows all substance to enter and to go out. Whereas what is the feature of this next one, that is cell membrane? Yes, it is semi-permeable, means only it allows certain substance to enter and certain substance to go out. So here outer one is called as what? Cell wall, and the inner one is called as cell membrane. So here root hairs have a thin cell wall, and you can see cell wall is freely permeable and cell membrane is semi-permeable in nature, and so only certain substance will be absorbed. So these are the characteristic features: huge surface area. Second one is cell sac containing higher concentration, and the third one is that cell wall is thin. These are the three features which enables the root for absorbing the water. Got it? Now you can see that absorption and conduction of water and minerals from the soil to the roots by 5 main phenomena is there. By main 5 processes taking place when water is absorbed by the roots to the other parts of the plant. Each one we can study one by one. First one is imbibation. The first term, what is called as imbibation. So what is meant by imbibation? It is the absorption of water in a living or non-living plant cells by the surface attraction. So the absorption of water by a living or non-living cell in a plant by the surface attraction is called as imbibition. You can see that substances which are made up of cellulose or protein, it has very high affinity towards water. So say for example, protein, you can see usually almost all seeds are rich in protein. So if this phenomenon that is called as imbibition, easily you can say that in your home and all, you can see that if you want to cook the dry seeds, your mother used to put it in the water and soak it in the water in the previous night. You might have seen, while going to bed, they will soak the seed in water completely, and then next morning when it is taken, you can see the seed will be swollen up. Why it is swollen up? By the absorption of water, because the main thing inside this dry seed is protein. So proteins have a high affinity towards water. Just like that, you can see that cellulose. Can you say material made with cellulose? Yes, wooden doors. You can see wooden doors during rainy season. It's bit difficult to close, or when once closed, you can see that these wooden doors will be bit difficult to open also. It becomes tight. Why it happens? Yes, it is due to this absorption of water. So this protein as well as cellulose, they have a high affinity towards this water, and so they are called as hydrophilic in nature. So they are called as hydrophilic in nature. So two examples also I told, dry seeds will absorb water. Even doors you can see that it become tight during rainy season by the absorption of this moisture. Okay. So again you can see that a pressure will be created due to this imbibition, and due to this imbibition pressure you can see seed cord ruptures. When this seed cord ruptures, yes, during the time of germination, you can see that you will put the seed in the soil. At the time of germination, you can see the radicle, plumule, all will be coming out. How? The seed cord want to break, isn't it? So, here also imbibition, we can see seed cord ruptures or breaks open due to imbibition. And next process is called as diffusion. So this is the second thing, that is absorption and conduction of water and mineral salts by the second process, that is called as diffusion. What is meant by diffusion? Diffusion is the free movement of molecule of a substance. It may be solute, it may be a solvent, it may be a gas. Okay. So, from the region of their higher concentration to a region of lower concentration. So, diffusion is the free movement of the substance. So, it is a solute, it may be a solvent, gas or liquid, from its higher concentration to a lower concentration. So this diffusion easily with an experiment we can prove. First you can see that what is this? Yes, this is a beaker. And what is this? Yes, it is a glass rod. So a simple experiment I am going to show here with this. So first thing is that I am pouring little water into this beaker. You have seen now in this beaker what is there? Yes, water is there. Now here in this beaker what is there? Yes, water. Here in this spatula I have taken little potassium permanganate. You see, now I am putting this potassium permanganate in this water. You can see clearly what is taking place actually in this process. Yes, that is diffusion. You can see slowly these colors spreading everywhere. So, actually potassium permanganate is a solute what I am putting in this water, and you can see now the color changes. When I am putting, you can see down here potassium permanganate, still some more crystals are there in here. So now what I am doing, so here the concentration is more where the crystals are there, whereas here the concentration is less. So solute concentration is less in this region. Now this is a glass rod. Using this glass rod, just I am stirring it. You see the color change? Yes, it became dark purple in color. Okay. This is a simple experiment to demonstrate diffusion. Once more, diffusion is the process by which the movement of the particle, that may be a solid, liquid or gas, that particle, from a higher concentration to a region of lower concentration. That is the definition. So when I put the potassium permanganate crystal first, it was down here. Slowly it started to diffuse to the lower concentration. What was there, which was lower concentration, it spread there. Then when I used this glass rod to stir that one, you can see that it is fully, you can see now the purple color came everywhere. So it is spread everywhere. So this is a simple experiment to demonstrate what? Yes, to demonstrate diffusion. Now I think all of you understood what is meant by diffusion. So where this diffusion applies, let us see. The molecules of the dye are more crowded and near the tablet of the dye. First, when I took this beaker, you can see that when I put that potassium permanganate, in that region the concentration was more, and the molecules move further and further. You can see the color spread. So it further and further, and you can see finally it distributed uniformly. Now we can see the third process, that is called as osmosis. Third process is called as osmosis. How can we define the osmosis? It is the movement of the water molecules from their region of higher concentration to a region of lower concentration. See the differences between diffusion and osmosis. Diffusion is the movement of the solute particle from a region of high concentration to a region of lower concentration. Whereas here you can see osmosis is the movement of water molecules. The solute particle, here water molecules from a region of high concentration to a region of lower concentration. And two types of osmosis takes place. One is called as endosmosis and the other one is called as exosmosis. So two types of osmosis, endosmosis and exosmosis. So in the case of endosmosis, that what endo means inward or to inside. So from the name itself you can see that this osmosis takes place to inside the cell. So water molecules this is into the cell. So water molecules get inside the cell, and so you can see the cell becomes swollen. So that process is called as what? Endosmosis. So endosmosis is the inward diffusion of water molecules. Now what may be the exosmosis? Yes, quite opposite to that one. That is outward. So the outward movement of these water molecules from the cell. When water molecules goes out of this cell, what happens is the cell will be shrinking in nature. In the endosmosis, you can see water enters, it will be bulged like this. Okay, and when water goes out due to exosmosis, it will be shrinking like this. Got it? These are the two terms connected with endosmosis and exosmosis. Now you can see that if the concentration of the surrounding solution and inside the cell, if both are equal, what will be the condition? Whether osmosis takes place, whether endosmosis takes place, exosmosis takes place, or no osmosis takes place. Yes, there is, because both the concentration are equal, so the osmosis will not take place. Now another experiment to prove osmosis. So all materials are not available here. So take some concentrated sugar solution in a thistle funnel. This is a thistle funnel. Okay, this is what is called a thistle funnel. Inside this, what you have to take? Sugar solution, concentrated sugar solution you have to take in this thistle funnel. And now what you have to do is cover the mouth of this thistle funnel with a cellophane paper. What is meant by cellophane paper? A paper which is semi-permeable. Afterwards, this mouth of this thistle funnel you have to cover using that cellophane paper or animal bladder or egg membrane. You can see that in the egg, that is thin membrane is there. That is also semi-permeable membrane, so that you can cover the mouth of this thistle funnel using an egg membrane. And now you have to invert. Okay, invert this inside this one, inside the beaker, you have to take water. inside the beaker you are taking water, and now here I have not covered, even I have not taken what sugar solution because cellophane paper is not available now. So just invert this thistle funnel is like this. What is inside this? Concentrated sugar solution. Here the mouth of this one is covered with cellophane paper and I am inverting it like this. Okay, and connecting to a stand. And keep it aside for few hours. Say for example 1 to 2 hours you can see. Before you are inverting, just after inverting, just to see what is the level of water in this tube. This tube you can see that level of water will be, say for example, this much. Just I marked with a chalk piece. Say for example this much. After 2 or 3 hours you can see that the level of the water will be increasing in this thistle funnel. How it will be increasing? Yes, you can see that sugar solution, it is a concentrated solution. The water concentration is less, whereas water from the beaker, you can see that water from the beaker enters through this thistle funnel and its mouth is covered with cellophane paper and which act as a semi-permeable membrane. So water enters through that semi-permeable membrane and so the level of the water will rise in the thistle funnel tube. So from this we can understand that osmosis has taken place and that's why the level increased. At the same time when you are conducting this experiment, you can this is called as experimental setup. Beside you can keep a control setup also, and in the control setup what you have to do, in the thistle funnel what you have to take normal water only, and here in the beaker also normal water, and then here the mouth of the thistle funnel you can cover with what? Yes, the same cellophane paper which act as a semi-permeable membrane and keep it aside. This will act as a control setup. So first one is the experimental setup. This is a control setup. So at the same time both you have to keep aside. So after few hours I have already told you in the experimental setup water level in the thistle funnel rises up, whereas in the control setup there is no change in the water level. What does this prove? Yes, because in the control setup both the thistle funnel as well as the beaker only water is present, whereas in the experimental setup you can see thistle funnel containing sugar solution which is more concentrated, that solute is there, that is sugar, more concentration is there, whereas outside water media is there the concentration of water is more. So you can see that the water as it is in a higher concentration, it enters from beaker to the thistle funnel where the concentration of water is less. So this is also another experiment that to prove this word osmosis. So from this I think you are clear about the definition of osmosis, that is osmosis is the process of the diffusion of water molecules from a region of higher concentration to a region of lower concentration. So you can see that here instead of this, here in the mouth of this thistle funnel you have taken what? Yes, cellophane paper. Instead of that you are taking the same experiment, just you conduct with what? A rubber sheet. In the first case, instead of this cellophane paper, you use a rubber sheet here to tie this one. And then same in the beaker, you keep it for 2 hours or 3 hours in this, inside this thistle funnel, sugar solution. Only difference is that the mouth of the thistle funnel is tied with a rubber sheet and you are keeping here. After 2 or 3 hours, can you observe any change? No, you can't observe any change. Why? Because you can say that the rubber sheet what we tied here in the mouth of this thistle funnel, it is impermeable. It won't allow this sugar solution either to go out or water to enter into this thistle funnel, as the rubber sheet what we type here is impermeable. Next case, instead of this rubber sheet, you have to take a muslin cloth which have many pores. So easily you can see that here when we are tying this muslin cloth, easily this sugar solution from this thistle funnel will go into the water which is present in the beaker, or water may enter into this thistle funnel as it contains many pores. So here three things we have taken. First one is a semi-permeable membrane. It may be, you can take an egg membrane or an animal bladder, you can take cellophane paper, you can take, all these are examples of semi-permeable membrane. So in the first case you have seen that the water rises up in the thistle funnel as it is semi-permeable, and moreover the concentration of the water in the beaker is more than that of the concentration of water what is in the thistle funnel, because it contains more salt, that is a solute. In the second case you have seen that you have taken a rubber sheet. This rubber sheet is impermeable, it won't allow the substance to go out or to in, so there is no change. And in the third case you can see that you have taken muslin cloth, their pores are present, and so easily from this you can see sugar solution will pass into the beaker. Okay. So here you can see that all these three cases, you can see you have taken sugar solution in the thistle funnel, but in the first case only you have seen that osmosis is taken place. In each experiment you have to keep a control setup, so easily you can understand the difference between this osmosis taking place and why osmosis is not taking place in the control setup. Now, how long this osmosis continue? So here osmosis in the first case, clearly you have seen this osmosis. And how long you can say this osmosis continue? Whether the solution in the thistle funnel, first case we have taken, in the thistle funnel the concentration is more, you can see that automatically water enters. So when it becomes an equilibrium, that is outer and inside, the concentration of the water becomes equal, you can see that osmosis stops. So that's all about osmosis which we have taken today. So what is meant by absorption? You can see that the process why absorption is needed, and this for photosynthesis, then for transpiration, transportation, and for mechanical stiffness plant needs what absorption of water, and these water and mineral salts are required by the plant for the synthesis of certain chemicals as well as for the enzyme. And what are the characteristics for absorbing water? It should have plant should have a large surface area. That means root and root hairs provide a larger surface area. Second one is that root hairs contain cell sap which are at a higher concentration than the water in the outer region, that may be in the soil. So you can see that this is nothing but osmosis. So cell sap was in a higher concentration, so that osmosis takes place, and so suction force will be produced and it helps in the absorption of water. Third one is that root hairs have a thin cell wall, and this cell wall which is freely permeable, and cell membrane is selectively permeable or semi-permeable, which allows certain substance to in and out of the cell. Now there are 5 processes or 5 phenomena by which this absorption takes place. 2 we have seen today. One is called as imbibition. Imbibation, it is the phenomenon by which living or dead plant cells absorb water by surface in attraction, and you have seen that seeds absorb water by this imbibition and it sells up. Again you can see the wooden doors and all. In the rainy season it absorbs moisture from the atmosphere and it becomes just little swells up, and so here also cellulose it is imbibing this water, and this protein as well as cellulose both are hydrophilic in nature. And again for the germination of the seed also you can see seed, if you want to germinate, plumule and radicle has to come out. So a force will be created and seed coat will be ruptured or open. That is nothing but due to imbibition force. And next one is diffusion. Diffusion is the free movement of molecules. It may be solute, liquid or gas from a higher concentration to a lower concentration region. You have seen the experiment with potassium permanganate. And the third one is osmosis. Here also you have seen it is a movement of water molecules from a higher region to a lower region. Hope that all of you understood. Thank you.